Hi, this is Shula from shulatutoring.com. Are you a student looking for additional practice, or maybe you are a science teacher looking for additional teaching materials that are free and convenient? If so, this channel is for you. So the way it works, I attached a practice test in the description for this video. So if you are a student, you will get the most out of this video if you go to the link and complete the test first. So you can either print the test and complete it this way, or you can just complete the test on a piece of paper while looking at the screen. So once you do that, you can watch the rest of this video with the explanation for the test. You can hear my explanation and score your points. You can see which questions you got right, which questions you got wrong. And this way you will be able to see if you are prepared for Mendelian genetics tests, like if you know this material. So make sure to add up your points at the end, and you'll see which grade you got. And for teachers, if you are a science teacher, feel free to use this resource whichever way you like. Just please keep the watermark, because that helps me spread my mission. So the test that I will complete as I'm explaining this material is in the Microsoft Word format. The test that you will download is a PDF, but the questions are the same. Okay, so let's start from the first question. In peas, round seed shape R is dominant to wrinkled seed shape R, and yellow seed color, capital Y, is dominant to green seed color, little y. Use this information to answer the questions below. For each genotype below, put check mark to indicate if it is heterozygous or homozygous. So homozygous means that you have two copies of the same allele. Here we have two copies of dominant allele, so this is a homozygous genotype. Here we have two copies of recessive allele, so this is homozygous as well. Here we have one dominant and then one recessive allele, so this is heterozygous, and this is two copies of dominant allele, so that would be homozygous. So, this question is 1 point each, so give yourself the points that you earned. Let's go to the next question. Complete the table by listing possible phenotypes: green, yellow, round, and or wrinkled. So those are all characteristics of seeds for each genotype. So it begins with the gene for the color, and then what follows is the gene for the shape. The first genotype, we have one copy of the dominant allele for the color. That's enough for that seed to be yellow. And the same with the shape. We have one copy of the dominant, so the seed is going to be yellow and round. So same here in terms of phenotype for the color. We have two copies of the dominant this time, so it's going to be yellow. But for the shape, we have two copies of the recessive allele. So that will make those seeds wrinkled. And again, we have two copies of the dominant allele, so that seed is going to be yellow as well. But this time, that seed is going to be heterozygous for the shape. So the shape is going to be round. And finally, we get some green seeds, right? Because we have two copies of the recessive allele. So this is going to be green. This seed is homozygous dominant for the shape. So it's going to be round. And let's see again, this seed has one copy of the dominant allele for the color. So it's going to be yellow. And the shape is going to be round because it's homozygous dominant for the shape. And finally, we have a completely homozygous seed for those two genes. So the color is going to be green and the shape is going to be wrinkled. Okay. So a plant for seed shape self-fertilizes to produce offspring. Complete the Punnett square below. So self-fertilization is when the plant is acting like the mother and the father at the same time. So it self-pollinates. So when a plant is heterozygous for seed shape, we have this situation, right? And because the plant is the father and the mother, then we have the same genotype for both parents. So we complete the Punnett square like this, and we get some interesting combinations. So some offspring will be homozygous dominant, and this is going to be heterozygous, right? So I'm bringing this guy here and this guy here, so vertically and horizontally, and then another heterozygous, and then one homozygous. So once we complete the Punnett square, we're continuing to describe genotype and phenotype of offspring. So here, offspring genotype, we have 4 different boxes, right? And each box represents 25%. So we have one box that's homozygous dominant. So it's 25% here. And then big R, little r, heterozygous genotype is 2 boxes. So that's 50%. And then we have 25% for homozygous recessive. Offspring phenotype for the round phenotype, it's anybody who has at least one copy of the dominant allele. So that's heterozygous and homozygous dominant genotypes. So that's 1, 2, 3. So 75%. You would need more space here. And then wrinkled, that's just 25% because you can only have wrinkled seeds if they have 2 copies of recessive allele. So let's continue to our true or false questions. A, the Punnett square above represents a dihybrid cross. So dihybrid cross is when we consider 2 traits. What we have here is a monohybrid cross. So that's going to be false. The only way for peas to have wrinkled seed shape is if they have 2 copies of the recessive allele. That's true. That's this situation right here. When pea plant that has green seeds self-fertilizes, 1/4 of the offspring will have yellow seeds. Well, green seeds are coded for by the recessive allele. So if a plant has green seeds, it means that it has 2 copies of the recessive allele, and when it self-fertilizes, it becomes the father and the mother. So we won't have anywhere for the dominant allele to come from, right? So 0 offspring will have yellow seeds, so this is false. When mating, a male and a female will contribute equally to their offspring, right? So like half of your chromosomes come from your father, half of them come from your mother. So that's true. All of the offspring of homozygous dominant plants will have round yellow seeds, regardless of the genotype of the mating partner. That's true because when homozygous dominant plants mate, they will pass one copy of their genes, one allele, to their offspring, right? And they only have dominant alleles here. So their offspring, regardless of the genotype of the mating partner or of the other parent, will end up with at least one dominant allele for those 2 genes, right? For seed shape and for seed color. So all the offspring will have round yellow seeds. Yes, so we just completed 2 pages of this test. So please make sure to score your points as we are moving along. So now some matching questions. Match each definition with the correct term by writing the corresponding letter to the left of each number. So version or copy of a gene, that's allele. Cross between a homozygous recessive individual and an individual of unknown genotype, that's test cross. So if you mate homozygous recessive with heterozygous individual, then some of the offspring will have the recessive phenotype. But if the other partner is homozygous dominant, like in this true or false question here, then all of the offspring will have the dominant phenotype, right? So that's what test cross is for. So moving along, genetic makeup of an individual, that's genotype. The idea that alleles separate during gamete formation, that would be law of segregation, right? E. Particular location on a chromosome, that's called locus, and plural would be loci. The way genes manifest themselves or traits that we can observe, that's phenotype. Phenotype that is most common in individuals in natural conditions, that's called wild type, right? So in Morgan's experiments, that's the word that you'll come across very, very often. The idea that copies of different genes are inherited independently from one another, and that's a law of independent assortment. Genes that are located on chromosomes other than X and Y, that's autosomal inheritance. A. Crossing individuals that differ in 2 traits, that's dihybrid cross. We use that one. So that's that. And then some multiple choice questions. In Morgan's experiment with fruit flies, some of the offspring had phenotypes and genotypes different from both parents. That could be due to what? So in Morgan's experiment, fruit flies had the genes under question on the same chromosome. And that's why they didn't exactly behave the way Mendel would have predicted. So that is due to crossing over, and that's what happens when homologous chromosomes exchange genetic material during prophase 1 of meiosis. The term linkage refers to, so this is actually a tricky question. A lot of times when you read your biology textbook, you will encounter words like X-linked or Y-linked or sex linkage. So that's different from just linkage. And it's something that a lot of students get confused about. So linkage, just linkage in itself, that's referring to genes that are residing on the same chromosome, D. It's not genes residing on either X or Y chromosome, because that would be phrased like sex-linked or sex linkage, right? So just be careful about that. Don't confuse it. In humans, individuals with AB genotype produce type A and type B glycoproteins on the surface of their red blood cells. This is an example of codominance. Those alleles, they code for glycoproteins that are found on the surface of red blood cells. If it was incomplete dominance, then those glycoproteins for heterozygous individuals would be something in between type A and type B glycoproteins. But in this case, the cell will produce both types. So that's codominance. The reason why Mendel used garden peas as his model organism is because peas have short generation time, they have characteristics that are easy to observe, and they're inexpensive. So all of that is true. In a rodent, brown coat color, capital B, is dominant to gray coat color, little b, but the presence of another G, capital A, is necessary for production of any pigment. So rodents that are all those are albino. This is an example of epistasis. Epistasis is when one gene affects expression of another gene. So that's another really important exception to Mendelian genetics. Short answer questions. A cross between a red-eyed Drosophila male fly and a white-eyed Drosophila female fly resulted in a red-eyed females and white-eyed males. Explain why there were no white-eyed females, and red-eyed males. We have to use the term X linkage. Let's just construct a Punnett square. So if you remember Morgan's experiments, you will remember that the gene for the eye color was located on the X chromosome. So what happens with males is that they have just one X chromosome, and females obviously have two. So a red-eyed male fly will have what is called the wild genotype, wild type. The female is white-eyed. So in order for a female to express sex-linked trait, if it's an X-linked trait, she needs to have two copies of it. So she needs to have that gene on both X chromosomes. So let's see what offspring they can produce. Here, the mother will give her X to make the sun, and the father will give his Y to make the sun. So this is not super clear, and I will just circle all the mutant alleles, right? So this is a mutant, this is mutant. It's supposed to be like a small w. So if you look at what's happening here, all the daughters that they produce have one copy of the wild type and one copy of the mutant type. So all the daughters are carriers, and the sons inherit their mother's X chromosome, one of them, right? And both of their mother's X chromosomes have that mutant gene. So all the sons are white-eyed. They are all affected. And this is just the nature of X-linked disorders, because boys have just one X chromosome, right? If they get the X chromosome with that condition, they will express it, because women have two. For women to express it, or for females to express it, they need to inherit two X chromosomes that both have that gene. So that's why males are affected by X-linked diseases at a much greater rate than females. Explain the difference between incomplete dominance and codominance. Well, incomplete dominance, you have heterozygous individuals will have intermediate phenotype. Like, for example, with the famous example of the flowers, if flowers have allele for white color and allele for red color, heterozygous individuals will be pink. And codominance, both alleles are expressed fully. Codominance, both alleles are expressed fully in heterozygous individuals. Compare and contrast pleiotropy and polygenic inheritance. So I really like this question because those things are direct opposites of each other, and it's a very easy way to remember them. So polygenic inheritance is when we have multiple genes, and it's easy to remember because poly is many, right? And genic is genes. Multiple genes that are affecting one trait. And if you can remember that, then you can remember that pleiotropy is the opposite. You have one gene affecting multiple traits. Polygenic inheritance, multiple genes affecting one trait. Pleiotropy, you have one gene affecting many traits. And finally, pedigree question. So we have this pedigree. The way it works, generations are numbered with Roman numerals, and then Individuals are numbered with regular numbers going from left to right. Squares are males, circles are females, and affected individuals are shaded. First question, is this trait autosomal or sex-linked? Well, if you look at the pedigree, you will see that only males are affected. So if you look at the pedigree and females and males are affected approximately at the same rate, then it's autosomal. If it's practically only males that are affected, then the trait is sex-linked. Only males affected. If the individual 3-6, where is 3-6? That's this individual. If the individual 3-6 gets married to a non-carrier female, what is their chance of having an affected child? So this is an X-linked recessive disorder, because we know it's not an autosomal disorder. Or it could be, it could be a condition of some sort. It's not necessarily a disease, but let's just say if that's true, the male that is affected by it has his only X chromosome with that faulty gene. Let's make a little Punnett square. So let's call that gene R and then Y. And then he is marrying female that's not affected at all. So just 2 X chromosomes. Let's see what kind of kids they can have. They can have XXR and then XXR. So if they have a daughter, the daughter will be a carrier. If they have a son, the son will get his X chromosome from the mother, right? So the son won't be affected at all and the son won't be a carrier. So that's a very important takeaway, that males cannot inherit X-linked conditions from their fathers, and they cannot be carriers, right? Because you can only be a carrier if you have 2 copies of something. And males, by their nature, they only have 1 copy of that X chromosome. So either they are affected or they're not, but they cannot be carriers. So their chance of having an affected child is 0%. Who is one person responsible for the origin of the condition in the family and why? Well, it's not showing in the first generation, but obviously this particular individual had to inherit it from somewhere. And because it's an X-linked disorder, it's inherited through X chromosome. And this is a son, and he got his X chromosome from his mother. So this individual is responsible. And bonus question, name one condition or disease that can be affecting the family in the pedigree above. So this is an X-linked disorder, a condition. So let's say hemophilia would be a good one. Color blindness would be a good one. Anything that's, that you can remember that's X-linked would work here. When you score your questions, you heard my explanation. So if you have something that's similar to my explanation, if you understand the logic behind my explanation, and if you have something like that written there, then just give yourself full points, or you can give yourself partial credit if you got to the answer partially, and you can just calculate your grade. One thing I have to tell you, every class is different. I had one student tell me, like, I was tutoring her the other week, and she was like, my teacher never mentioned epistasis. I don't think we need to know it. Whereas epistasis, I had a question about it. So if you don't need to know something from this test, just subtract those points from the total grade. It's like this test is 100 points. For example, multiple choice questions are 3 points each. So subtract those 3 points and calculate your grade out of 97 points. So now it's time to calculate your grade. If you got a good grade, congratulations. If your grade is not so good, good job for trying, but at least now you know which areas you need to focus on so that you can really, really master this material. So if you enjoyed watching this video, please give me a like. If you have any questions, please mention your questions in the comments below. And I'll be coming out with more videos like that. I'll be making more practice tests on different topics in biology and chemistry. So please subscribe to my channel so that you don't miss future content. See you soon. Bye bye.